Right, thank you for the introduction. I'm here from the HABS faculty and in the School of Human Movement and Nutrition Sciences. My working title is Training to Enhance Neuromuscular Control of the Ankle in Cerebral Palsy. So cerebral palsy is a neurological condition. It results from an abnormality or damage to the brain around the time of birth, which results in impairments of movement and posture. Within the lower limb, this greatly affects the capacity to walk, particularly the ability to lift the toes and clear the ground, as you can see here with the right toes dragging along the treadmill belt. So my question is, does active movement training improve neuromuscular control of the ankle in cerebral palsy and transfer to functional tasks? Active movement training is that which requires volitional muscular effort to produce force or a joint position change. And neuromuscular control describes the integration of the nervous system, the brain, and our muscles to produce movement. So what are we dealing with? Well, cerebral palsy results in motor impairment, and this is greatest at the ankle, due to the brain abnormality occurring proximal to these nerve pathways to the ankle. This results in muscle adaptations. So as we can see here, there is reduced muscle volume in the left calf and an equinus foot posture. The heel cannot touch the ground as there has been shortening of muscle fascicles and tendons. Now, traditionally, muscle is the target of exercise interventions, yet this does not address the root cause of the muscle adaptation, which is due to the disruption of the brain. There are neural adaptations. So a few things we do know is that the rate of electrical signals that are being sent to the muscles is reduced. There's variability in the timing of muscles contracting and relaxing, and the capacity to voluntarily activate them is severely reduced. Yet the neural mechanisms behind these changes remain poorly understood, and the capacity for the damaged brain to learn to improve ankle control has not yet been investigated. So there is promising evidence that active modalities can improve neuromuscular control. Cross-sectional studies show a reduced ability to voluntarily activate muscles within the lower limb as much as 20 to 50% in cerebral palsy. However, this has not been assessed post-training in this population. So research has shown that following resistance exercise in healthy populations, this capacity to voluntarily activate muscles can be improved. As I've already mentioned, one of the greatest challenges for individuals with CP is tripping due to reduced ability to dorsiflex or lift the toes, as you saw before. Explosive resistance training in the lower limb has been shown to improve the rate that muscles produce force, as well as the extent of toe lift during walking. Even daily walking has been shown to improve the amount of toe lift achieved, as well as coherence in the muscle that is responsible for this movement in the cerebral palsy population. So whilst all these papers show some favourable and very positive findings, the neural measures, if they are performed, are done so in isolation, and they're using resistance training to target muscle adaptations and not the brain, which is the root cause of the movement impairment. So I intend to implement a novel motor training intervention based on motor learning principles, where participants are required to do two tasks. One, moving the ankle, and the other with the ankle in a fixed position, applying a force to trace along a line. So participants will be presented with this black line, a random trace, and they will have to move their ankle position to match it as accurately as possible. So the expected ankle position is shown here in red, and participants are given an accuracy score following each trial. So this training will be performed for 6 weeks, 3 times weekly, 30 minutes of training. And it's hypothesized that this active movement training will improve ankle position control and joint movement accuracy, which will in turn increase ankle power production and the ability to lift the toes while walking. To test this hypothesis, I will recruit 20 individuals aged 15 to 25 with spastic hemiplegic type cerebral palsy. Testing will occur across 3 time points through 3 different domains. Neuromuscular control will be assessed by recording electrical muscle activity during electrical stimulation and strength measures, which will allow me to assess changes in neural activity and function. which is currently not understood. Selective motor control measures have been included because they are directly transferable out of research and into clinical practice for use by allied health professionals to assist and assess rehabilitation. Function will be assessed using 3D motion analysis, force plates, and musculoskeletal models to see if the motor learning will enhance walking, specifically that ability to lift the toes, to produce propulsion, and for gait speed. A single group acting as their own controls will be used, as this has been shown to reduce variability in heterogeneous samples, which the condition of cerebral palsy is. Recruitment will be primarily sought through the cerebral palsy register. In addition, social media and contacts with rehabilitation clinics will be utilised. I currently have extensive experience with the techniques and the equipment that will be used, and we have this on site in our lab. In addition to this, I've been developing skills in building custom software and an equipment prototype, along with learning new specialised analysis software and the statistics required to carry out this project. So what's new? This study will use a novel training task which is specific to the control required to walk. It targets the brain, that is, which is responsible for the motor impairment observed within this neurological condition, and not the secondary adaptations that occur to muscle morphology, which all the previous literature has investigated. Previous studies that do use neural measures do so in isolation. So here I will use a combination of neural assessments to gain a greater insight into the potential mechanisms possible for the intervention outcomes. So what? Well, these individuals have reduced walking ability and trip over their own toes due to reduced ankle function, and this can worsen throughout life. This study will provide crucial information on how this type of novel motor training could maintain or improve neural function at the most impaired joint, the ankle, as we don't currently understand the neural mechanisms behind their impaired movement. So this study will contribute to a greater insight into neural mechanisms of motor impairment within the lower limb. Positive outcomes may contribute to the development of new, effective and targeted rehabilitation protocols which can be implemented not just in children but in adolescents and adults, in order to improve neuromuscular control and therefore walking ability in these individuals with cerebral palsy. Some additional considerations. The equipment I'm building will be portable to increase accessibility and participation in this study. The risks are deemed to be low as the measures are non-invasive and require the participant to produce voluntary muscle activity and movement, and all sessions will be supervised. There is a low risk of no result, as improvements in this training task are expected given what is known about the principles of motor learning in humans. There is low to nil risk that this research is obsolete, due to the fact that minimal data has been published on neural assessments following active training modalities in cerebral palsy. And in addition, that this condition affects so many individuals. It's the leading cause of childhood disability in Australia and is present throughout their life. So there is a high demand for effective neurorehabilitation. In summary, this project seeks to establish how the nervous system, which was disrupted at birth, can learn to improve neuromuscular control of the ankle and subsequently walking ability. Thank you very much.